Alright guys, so this video is going to finish up the lesson on viruses and bacterial genomes. And this is going to be relatively short. We just have a few slides left to cover. And hopefully you know some of this already because it is going to be a review of things that we covered in first semester. So before we really kind of get into it, I want to talk about a couple of things. Okay, number one, once again, we see the term genome, and we need to remember that when we're talking about the genome, we are talking specifically about the genetic material. So all this portion of the lecture focuses on is just the genetic material of bacteria. And just another reminder here, bacteria are prokaryotes. So this is why we should know some of this already, because we know that the genome in a bacteria may be slightly different than a genome in a eukaryotic cell. Now, once again, why are we talking about this? Well, we're going to get into DNA technology, and bacteria are used very heavily in DNA technology. So you should have already watched the video about how DNA technology came about. And one of the things you saw was that at the Genentech, we're using bacteria and cells to make human drugs like insulin. And this is something that has really changed pharmaceuticals. So the reason we're talking about viruses and bacteria is that these two things are really used very commonly because of their genome and because of our ability to kind of manipulate. We use them quite a lot. We don't see a lot of your genetics using, large mammals and things like that, because a lot of times there are issues in using those organisms in science. But we can use bacteria in just about anything we want to, and it's okay because bacteria divide rapidly. No one's going to be upset if we kill a few bacteria along the way, and so they are used very heavily, especially in DNA technology. And so what this particular portion is focusing on is just the genetic material of bacterial cells. Now, I do want to remind you that the first portion was about viruses. Viruses are not living. Viruses are not cellular. They don't carry on all of the activities of a living thing. They are pretty much genetic material surrounded by protein. And that genetic material can vary. It can be DNA, it can be RNA. Now, here we are talking about bacteria, and bacteria are living cells. They are prokaryotic cells, and so they have all of the characteristics of life. They have cellular structure. They have a cell membrane. Most of them have cell walls on the outside. They have cytoplasm. And so I don't want you to get these two things confused, okay? Viruses not living, bacteria living. So in terms of their genome, we know hopefully already that they do not have a nucleus, so their DNA is found in the nucleoid region, which is just wherever it is floating in the cytoplasm. Now, I want to remind you too that DNA in a living thing always has the same structure. If it is in a living cell, DNA is going to be double-stranded, sugar phosphate backbone, nitrogen bases paired up on the inside, A to T, C to G. Now, I'm not going to say that there are 0 exceptions to the rule, but in general, if we're talking about a living cell, the DNA has the same basic structure that we learned about last semester. Now, so I don't want this to confuse you, okay? When it says single, okay, we're talking about one chromosome. All right, so remember that in our cells, we have 46 chromosomes, and they look like X's. We call those linear chromosomes. But the DNA in a bacteria kind of wads up to form one circular chromosome, okay? Now, that DNA is still double-stranded. That DNA has the same structure. But for whatever reason, this word always throws students off. So when we say single, we're referring to a single chromosome, not a single strand of DNA, okay? So it's a double-stranded DNA coiled up to form a single circular chromosome. Now, in our cells, in our DNA, we have those non-coding regions called introns. Bacteria really don't have a lot of these. Pretty much the information that they have is going to contain important information. Now they are going to have an origin of replication, and this is going to be important in understanding DNA technology and making recombinant DNA, because in order for that genetic material to get copied, it has to know where to start. So there's a location on that circular chromosome called an origin of replication, and we're going to see that coming up again. Now, in addition to their single circular chromosome, which is going to have essentially the information needed to be a bacteria, they may also contain these smaller circular pieces of DNA called plasmids. And this is a term that you need to get pretty familiar with, because in terms of DNA technology, these plasmids are used pretty heavily. Okay, so they're smaller circular pieces of DNA. They can replicate themselves. Okay, they're going to have an origin of replication just like the chromosome. But the thing is they are disposable. Okay, they aren't essential to be a living, working bacteria. So up here on the chromosome is going to be all of the genes needed for that bacteria to sustain life. But there could be some extra genes found on these plasmids that could give that bacteria some additional traits. Okay, and we're going to look at some of those. Now, what the plasmids might contain are genes that allow it to survive better. Okay, they don't necessarily have to have them. And one of the important things to know about plasmids is that they can be exchanged. So plasmids can be moved from one bacteria to another. They can be taken in from the surroundings. And so anytime a bacteria were to take in these plasmids, they then will take on the traits that those genes possess. Okay, so a couple of examples are F plasmids. I'm not sure where the F comes from, but F plasmids allow bacteria to conjugate. And we're going to look at this term here in a minute. So if bacteria contain the F plasmid, we call them F plus or F positive. If they don't contain that plasmid, we would call them F negative or F minus. And so what this does is it allows the ability to conjugate. Now, conjugation is the ability of bacteria to form a hollow tube. They can connect it to another bacteria, and then they can exchange these plasmids through that hollow tube. Think of it kind of like a bridge. Now, if they do not have the F plasmid, they cannot form that little hollow tube. They can't form that bridge. But if they do have it, they're able to conjugate, form that little empty tube, and pass these plasmids from one bacteria to another. Now, another one is an R plasmid, and this one's going to be important because we see these used a lot in DNA technology. And the R plasmid, the R stands for resistance, and they contain genes for antibiotic resistance. Now, I know that you may be thinking that antibiotic resistance is a bad thing. For us, in terms of our health, we do not want bacteria to be antibiotic resistant because that means we can't treat them. However, when it comes to DNA technology, this is one way that we can identify bacteria. Antibiotic resistance is going to be a very important, what we call a selectable marker of how do we know the bacteria got the information. And so what we can do is we can use this to determine which bacteria received the plasmid and which did not. So we'll talk about that more later. For now, just understand that plasmids are these extra smaller circles of DNA that you'll find in bacteria. They may give some additional traits such as the ability to conjugate or antibiotic resistance. All right, now this right here should not be new information to you. Okay, so when it comes to reproduction, remember that bacteria are asexual. They divide by a process called binary fission, and they can reproduce very quickly. Okay, they can demonstrate some exponential growth, which means they're essentially doubling. Now, I don't know that this is fully understood. We did talk about like mitosis in eukaryotic cells. It's going to be somewhat similar to that, but without the nucleus stuff. And so essentially the DNA is copied in the bacteria. It starts at that origin of replication, and then the rest of the cell is going to be divided. And remember that the key is you get the same. Because it's asexual, you're going to produce identical bacteria. Now, that does not mean that there can be no diversity in bacteria, because there are other ways that diversity and variation can be generated. It's just they're not generated through the basic reproduction process. And so there's 4 other ways that you can generate variation in bacteria populations. Now, the most common is going to be just simple mutation. Okay, remember that anytime we copy our DNA, changes can be made. And so because of that very high rate of reproduction, there will be mutations, okay? DNA replication is not a perfect process in prokaryotes or eukaryotes. And so one way that they can generate some changes is just through random mutation. And like I said, that's going to be your most common way to generate some variation. Now, there are 3 other types of diversity generating processes, okay? So these are ways that DNA can be altered in bacteria. And all of these are going to involve getting genes from another bacteria, okay? So all of these are examples of gene exchange, okay? And what you need to understand is where are they getting the genes from, or how are they getting the genes? Now, these 3 terms here you will see again. When we cover prokaryotes later in the semester, we're going to go through each of the kingdoms, and we're going to talk about organisms on a larger scale. We're going to see these terms again, okay? So you've got transformation, transduction, and conjugation. And transformation is going to be especially important in DNA technology. So transformation is just the uptake of foreign DNA. And so what can happen is bacteria can take in DNA from their surroundings. They're literally able to bring it through their cell membrane. Their membranes become kind of weak and leaky, and they are able to take in DNA from their surroundings. Now, you know those bacteria that are making human insulin? They are transformed, okay? We can create bacteria by transformation, by transforming them. And so this is just anytime you take in foreign DNA from your surroundings. Now, the experiments involve making bacteria more susceptible to transformation. We actually used to do a transformation lab in DC Bio, but we don't have enough time with our 45-minute classes. But we're going to see this one coming up again in DNA technology, and this is definitely a concept to understand. Now, transduction, now remember that a bacteriophage is a virus that infects bacteria. Transduction is where viruses are transferring genes from one bacteria to another. And so as they go on to infect new bacteria, they could transfer some genes from previous bacteria. So transduction is when viruses transmit genetic material. And then conjugation is like what I mentioned earlier. To be honest, it kind of looks like bacteria sex, but I want to remind you that bacteria reproduce asexually. So this is not a reproductive process. This is just a way to kind of shake up the genome. And so one bacteria, if they have that particular gene, that F plasmid gene, they're able to create what's called a sex pilus, okay? Now, pili are just little extensions that can be found on the outside of bacteria for sticking, but there's a modified pilus that creates that hollow tube. It's called a sex pilus. And so if the bacteria has the proper genes, they're able to form that sex pilus, and then they can pass plasmids from one bacteria to another, okay? So once again, through reproduction alone, bacteria do not generate diversity, but by mutations or gene transfer, they are able to shake up the genome a little bit. So transformation is taking it in from the surroundings, transduction is a transfer by viruses, and conjugation is an exchange of plasmids through a sex pilus. Okay, so these are definitely some terms to know. So over here on the left, we have a picture of binary fission. Once again, you have an origin of replication. The DNA is copied, the bacteria divide in two, and they can do this very rapidly. Over here is a picture of conjugation. Okay, so here's my chromosome, here's my plasmid. If the bacteria is able, they can form this hollow tube, this sex pilus, and they're able to connect to another bacteria, and that pilus can transfer a plasmid from one bacteria to another. Now, once that plasmid, remember they can replicate themselves, so once that plasmid is transferred, then this bacteria would now have the ability to conjugate. Okay, so it's not a reproductive process, it's just a gene exchange. All right, the last thing we're going to talk about is gene expression. And we know from eukaryotes that gene expression is highly regulated. It's going to be regulated in prokaryotes as well. But because they just have that one kind of circular chromosome and maybe some plasmids, their gene expression is going to be slightly different. Prokaryotes have what we call operons. And you're actually about to watch a video as soon as this is over and answer some questions, and he's going to be able to explain it better than I can. But an operon is just a method of gene regulation in prokaryotes, okay? So remember, they don't have a lot of chromosomes. They just have one, and they don't have all of that extra fluff DNA. So the genes that they have are all kind of right there together. And so essentially what an operon is, it's a group of genes. So a lot of times to accomplish a certain task, you may need several proteins which come from several different genes. And so an operon is a group of genes that are either all turned on and expressed together, or they're all turned off and not expressed together. This is a little bit different from us because in our chromosomes, for the most part, every gene has its own promoter. Okay, now this is taking us back to first semester. So I just want to remind you that a promoter is where RNA polymerase binds. So when we're getting ready to copy our genes in transcription, RNA polymerase would bind to the promoter, and then it would go on and copy the gene. Okay, so this is just a sequence of DNA that tells RNA polymerase where to bind. And so in our chromosomes, each gene has its own promoter. So we copy these genes individually as needed, okay? Once again, it's highly regulated. But if you had a gene for a particular enzyme, it's going to have its own promoter. A gene for a different enzyme would have its own promoter as well, okay? Now, how that's different is here we have one promoter for multiple genes. So we have this series of genes. So there's three main parts to an operon. You have an operator, which think of that like an on-off switch. It's going to regulate whether the genes are copied or not. And remember, typically if we copy them, we express them and make that protein. You have the promoter, that DNA sequence where RNA polymerase binds, and then you have the series of genes that they control, okay? And the video is going to cover this again as well. So these operons can be regulated by regulatory molecules, and most of them are what we call repressible operons, and that there are repressors that are going to inhibit transcription. So remember, if you repress something, you slow, stop, you hold back, okay? So repressors are proteins that bind to the operator, and essentially they halt transcription. So repressors means we're not copying the genes, we're not making the proteins. Now there's some other regulators like co-repressors. They're going to help out the repressors, which means essentially the genes are going to be turned off. There's inducers, which inactivate the repressors, and then there's activators, which help RNA polymerase bind. So inducers and activators are going to help promote transcription, which means they're going to kind of help turn the genes on. Repressors and co-repressors are going to stop transcription, so they're going to kind of turn the genes off. So there's a couple of examples, and both of these are going to be discussed in the video, okay? But there's two examples that are commonly found, and you've got the trip operon and the lac operon. Now, you'll find the trip operon in E. coli. Those are just some common bacteria. They're used a lot in genetic study, and they're used a lot in DNA technology. But these bacteria can either ingest, which means they can eat, or they can make tryptophan. Now, tryptophan is one of your amino acids. It's actually the chemical tryptophan is what's in turkey that they say makes us sleepy at Thanksgiving. But essentially, you have to either ingest or make amino acids in order to build your protein. So these particular bacteria, they can either eat it or they can make it. Now, it's kind of about conserving resources. Why would you make it if you could just consume it from your surroundings? And so if tryptophan is present in the environment, then what happens is that kind of stimulates the repressor, and those genes are turned off because there's no reason to make it if you can ingest it. Now, if that tryptophan is not really in the surroundings, or it's very low, it actually causes the repressor to unbind, and that allows the genes to be copied so that they can then make tryptophan. So it's usually just about what's in the environment, okay? So the tryptophan is going to usually bind to proteins, and those proteins can bind to other proteins. It's all part of that cell communication, very specific. But essentially, it's turned off if tryptophan is available. It's turned on if tryptophan is not available. And that way they can use those genes to make the proteins needed to make tryptophan. So this kind of shows you the promoter. Remember, this is where RNA polymerase binds. The operator is where that repressor binds. So notice that if tryptophan is available, it binds to the repressor, which then binds to the operator, which means these genes are not going to get copied, okay? But if tryptophan is not available, the repressor is not bound to the operator, which means these genes will get copied. All right, and the last example is the lac operon. Okay, so this is an operon that involves activators as well. Now, once again, it's usually about conserving resources. So just like us, glucose is a common sugar that's used for energy. And so in these particular bacteria that have this operon, if glucose is present, then they're going to use it and they're going to break it down. But if it's not present, they can use other sugars like lactose. Now, once again, why make the genes or why make the proteins that you need if you don't need to break down lactose? Okay, so it's all going to be about what's present and what's not. So in order for the operon to be activated, there can't really be glucose a lot in the environment, because if glucose is there, they're going to use glucose. So the glucose would have to be low, but there would also have to be the presence of lactose. Okay, so once again, if there is plenty of glucose, that repressor is going to be bound, and there's no reason to copy the genes to digest lactose. If lactose isn't around, that repressor is going to be bound, and there's no reason to copy those genes. So in order for those repressors to not be bound or the activators to bind, glucose must be low and lactose must be present. And so it's all about the conditions, and that will determine whether or not the repressor is bound or not. And ultimately that will decide whether the genes are copied, okay? What you really need to understand more than anything is that an operon is a mode of gene regulation in prokaryotes. It involves a single promoter and a series of genes. And either all of the genes are turned on, which means the repressor is not bound, or all the genes are turned off together, which means there's a repressor present, okay? So here in a minute, you're going to watch a video, answer a few questions. It's just, it's very short. It's only 10 minutes long. And so that will hopefully help you understand operons a little bit better. So you will have a quiz coming up on viruses and bacterial genomes. I'm sure Miss Petty's going to tell you this, but it is a true-false quiz. So just make sure that when you're studying in preparation for recognizing statements as either true or false. It is not meant to be tricky. It's just to make sure that you understand the material. And so it's not a definition type quiz necessarily, but you just need to understand the material enough. They should be very clearly true or very clearly false. So once again, it's not meant to be tricky. It's just the way this quiz is going to be formatted. So just be sure you study your notes, ask any questions if you have them, and I hope you guys have a great day.